So after they use the single-cell sick fish and the C1 smart fish to answer these two questions, they are going to the next step, use the classical RNA-seq to digest the transition more precisely. So firstly, they use the UMAP and the t-SNE analysis to analyze the 10x chromin data, which is the whole transcriptome analysis. So the UMAP analysis can easily dissociate the cells into two groups, which is the DN3 groups, and the ETP-DN2, which is a huge mixed group of both progenitor and the transition stage. And the t-SNE analysis further review a new cluster, which is unknown cluster, and which consistent with the sick fish data and the C1 smart sick data. And if we map all the marker of the T cell lineage back to the t-SNE data or UMAP data, we can find they are distributed at different locations of the map, indicate that the cell can be easily identified by the different markers. And here is the very interesting result, because when we use like the granulocyte or macrophage or NK cell to label the population, we can also find they are restrictedly expressed at a certain area, such as here. So that's leave the possibility that maybe this small cluster of the cell are non-T cell that can contribute to either the granulocyte or macrophage or NK cell. After the group obtain the sick data from the 10x chromin, they firstly do the unsupervised clustering to see whether they can automatically cluster the cell into several groups. And they have successfully detected the ELANE expressing outgroup, which is the cluster 13. And we can find that here is a cluster of the genes including ELANE, PRTN3, and LY6C2. That is non-T cell lineage markers. And also this cluster of the cell expressed a totally different transcript from any other clusters. And also they find the stem cell cluster which expressed a lot of progenitor genes, and also the group 9 and 11 that expressed a lot of end stage of the T cell lineage markers. But except for these clusters, they also find a lot of clusters that expressed some early or late markers of the T cell lineage. These clusters have the greatest interest because they representing earlier pre-commitment pro-T cells, but the ordering was ambiguous because the T cell key genes such as the GATA3 and the TCF7 did not change sharply, and several substates associated with the cell cycle genes. So here comes two questions. The first question is that, as we generally know, when we do the differentiation or developmental study, we know that there are several markers, they will appear or disappear. And the expression pattern is consistent with the differentiation of the cells. For example, if one cell go into the differentiation process, their lineage-specific genes will increase. So if we follow the expression pattern of these genes, we can find the substate according to the increased tendency. So that comes to the question how we can follow the expression pattern of the GATA3 and the TCF7 to digest the stage in an correct order. And the second question is that why they're always correlated with the cell cycle genes. How can we use the cell cycle genes to determine which one is more mature and which one is less advanced? The general developmental knowledge is that before the cell enter the mature step, they have the higher capacity of cell renewal. But after they go into the differentiation stage, then their proliferation ability will be restricted. So here it's very hard to use the cell cycle gene to determine which stage is less mature. So to answer these questions, the group firstly trying to find out the outgroup of the cells that has been reviewed by 3 different methods. So these groups, they trying to validate their non-T cell properties by the in vitro cell culture. They're trying to isolate the cells from the side mode again, and they use 2 different markers they identify from the cluster, which is the CD33 and LY6C2. So here is the first novel findings they have. After they use the CD36 and the LY6C2 to isolate the cells from the ETP population, they culture the cell for 4 to 8 days with both the T cell induction medium and the non-T cell induction medium. Here is the flow cytometry analysis result. They use the CD25 as the marker to evaluate whether the cell can go into the fate of the uncommitment DN2 cells. And here is the double negative cell population, which should be the T cell lineage. And almost all of them can enter the cell fate of T cell lineage. But the double positive marker of the cell fail to enter the T cell lineage, which is indicated by the negative of the CD25. So that means this population, the LY6C positive CD63 positive cells, is not the precursor of the T cells. Instead, they try to use the granulocyte medium to induce these two populations, and they find the double negative cells fail to enter the granulocyte fate, which is indicated by the marker GR1. And when they try to culture the double positive cell population, and they find most of the cells successfully enter the granulocyte cell fate. So by this in vitro cell culture, they successfully validate that the non-T cell lineage group, which is indicated by the single cell sequencing before, is the precursor of the granulocyte. Then they try to answer how to order the cluster into a correct order that follow the trajectory of the cell development. So they use two analyses to answer these questions. And the first analysis is called spring analysis, which show the actual trajectory of the cell take. So what is the spring analysis? This is analysis that can visualize the long distance and the nearest neighboring relationship of cells across 3 reduced dimensions. For example, if 2 cells are very similar to each other, according to their transcriptome data, their distance in this map should be very close, so they're neighboring cells. But if there are 2 cells that are very different from each other, such as the progenitor and the end-stage T cells, then their distance is very long and they're far away from each other, such as the cell here and here, or here to here. And according to the spring analysis, the cells sequenced by the single cell sequencing are distributed in the 3D dimension maps, such as here. And then this group trying to map all the markers back to the 3D models and to see how they're distributed in this model. When we look into this map, we can find there are 2 axes. The first axis is from the early genes to the late genes. How do we make this conclusion? So all of the genes here are the progenitor genes of the T cell, and they are all distributed in the upper right corner of the map. And here is the late genes of the T cell lineage, and they are all distributed in the left bottom corner. So here is the axis, the first axis of the T cell development from this angle to this angle. This is the early state and this is the late state. And here is the second axis, which is from here to here. So here is the cell cycle that indicated by the SAMHD1. And here is another angle that indicated by these 3 markers. So the first markers is like a progenitor marker, and the left 3 markers is like the proliferation marker. So the second axis is like from the progenitor to the proliferation. And if we look into this 2 axes, it's more like the cell transitions through multiple cell cycles as they progress through the successive differentiation state, rather than confining cell cycling to a single state. And how do we interpret this sentence? So according to the general knowledge we have mentioned before, when the cell entered the differentiation stage, then they lost the ability to proliferate. that's a common sense. However, here we can find when the cell going from the less mature to a more mature stage, actually every stage the marker are overlapped with the proliferation marker. So it's more like that every time when they enter the next stage, they will proliferate for several times and then give to the more mature cells. So when they do the cell transition, they also do the cell cycle all the time, but rather than they do the cell cycle at first and then sharply enter the differentiation stage and not divide anymore. And also we can find that here are some non-T cell lineage marker expressed at a different stage. So that means maybe there are also some markers that indicate the non-T cell lineage appearance. And here is the GATA3 and the TCF7. They are expressed almost everywhere. So that means in the early and the late stage, they are always there and overlap with both the stem cell and T end cell lineage. And that suggests that they are more like genes that can connect the two different regulatory networks. So if we look further into the spring analysis model, and then we can find that they show a very interesting trajectory the cell takes. So when we see the expression pattern of different markers, and we can find some of them are not overlap, such as the FLT3, which is the immature markers of the ETP, and the BCL11B, which is the marker of the mature DN2 cells. So these two are non-overlap completely. But here is another markers that is very interesting. The BIRC5 actually indicate the G2 cell cycle phase. So that means the cell is still in the cell cycle and do the proliferation. But this population actually include both the immature ETP and the mature BCL11B or the IL2RA. So that means both FLT3 and DN2 cell population are endure proliferation. And the second finding is here. It's very interesting because the SPI1 is the marker for the T cell lineage, but the CD7 is a non-T cell marker. But why these two populations are overlapped? Whether that means here have some non-T cell lineage, or the CD7 is a transition stage marker that indicate the cell are priming for the next stage. So here comes to three possibilities for this overlapping phenotype. The first one is the developmental branching point that the cell, the progenitor cell, can give to both T and non-T cell lineage, or an alternative entry point for precursor. So as we know, the FLT3 is priming for the next stage of the DN2A and already entered the T cell fate. And here may be the second entry point that the SPI1 cell is also prepared for their next stage for the DN2A cells. And the third possibility is that the transient induced upregulation of the non-T genes even along the T cell pathway. So that means even though these genes are not expressed in the end stage of the T cell lineage, but actually they're the marker of the transition stage of the T cell development and contribute to the transient wave of the gene expression pattern.